Okay, so this presentation is about Frederick Griffith and how he helped to discover that DNA. Alright, this presentation is about Frederick Griffith and how he helped to discover that DNA was the material that carried our genetic information. It's going to cover objective DNA 1, summarize the experiments that led to the discovery of DNA as the genetic material. So in summary, 1928, Frederick Griffith used 2 strains of bacteria to study how a factor or a trait could be transferred from one species to another, or from one strain of bacteria to another. And the 2 strains of bacteria he used were the same, except that one had a gene that allowed it to make a capsid, or a protective coating around it, and that was the smooth strain. And the smooth strain that forms that capsule protected the bacteria from the mouse immune system. So the bacteria could cause pneumonia and then kill the mice. The rough strain doesn't form the capsule, so the mouse immune system can kill that type of bacteria. So in Griffiths' experiment, he used the rough strain, the safe one, and injected it into the mouse, and the mouse lived. Then he used the smooth strain, injected it into a mouse, and that mouse died. And that's what we expect, because we know that's what the bacteria does. So then he heat killed the smooth strain, so that bacteria was dead, and injected it into the mouse, and the mouse lived. So he showed that the heat-killed smooth strain was safe for the mouse. So then he mixed the 2 safe ones, the rough strain, the living rough, and the heat-killed smooth, both individually safe for the mouse. But when he mixed them together, he injected the mouse with that, and the mouse died. When he tested this mouse's blood, he found that there was living smooth strain bacteria in the mouse, even though he hadn't injected that mouse with any of the living smooth. So Griffith concluded that there was something in the dead smooth bacteria that was transferred to the living rough bacteria when they were mixed together. And he hypothesized that whatever it was that was transferred there transformed the living rough into living smooth. His evidence for this was that when he mixed 2 safe samples of bacteria, the living rough and the dead smooth, and mixed them together and put them in the mouse died. So that was one piece of evidence. The other piece of evidence was that he found living smooth bacteria in those mice.